circulatory system. The first spot that it is encountering is the liver, where it may or may not be metabolized by first-pass metabolism enzymes. The remaining drug circulates throughout the body and can distribute up to the CNS. Now, if you can get it across the blood-brain barrier and distributing through the tissue to your target in sufficient quantities, you're in a good position to have some efficacious response. On the right here, these are a couple experimental measures of how we quantify CNS penetration. So log BB is one term you might hear quite frequently. This is simply a comparison of the drug concentration in the brain relative to what you're seeing in the plasma. You might also see the term %F, which is a relative bioavailability measure. The key thing here to remember is that molecular properties of the drug are really what's influencing how the drugs are absorbed in the body, how they're distributed, how they interact with transporters, and even how they get metabolized by the CYP or CYP enzymes. And just a couple examples of how CNS bioavailability can impact things. I like to use an example of antihistamines. So this isn't exactly a CNS drug, but I think it kind of illustrates the point here. On the left, you have first-generation antihistamine, and a lot of these have been characterized by sedative side effects, so CNS pharmacology that would be undesirable. As second-generation antihistamines were developed, they were able to design compounds that lacked that drowsiness feature. So, interestingly enough, the compounds in the second generation that lacked drowsiness ended up being P-glycoprotein substrates, so they were effluxed back out of the CNS, whereas these first-generation compounds were brain penetrant and avoided efflux. So kind of a counterexample, if you will, but it illustrates that CNS bioavailability can have different effects. Now, I also mentioned CYP2D6 or CYP2D6 as a case study, simply because of its importance in CNS drug metabolism. Now, if you've heard about drug metabolizing enzymes, 3A4 is an isoform that generally garners a lot more attention. And if you examine drugs overall, it is more involved broadly. But if you key in on just CNS drugs, 2D6 is actually involved more broadly there. So another complicating factor with 2D6 is that you have genetic polymorphisms that affect its expression and function. And what that means is you can have variable drug metabolism. So you have an example of a cancer drug on the left here, tamoxifen. This gets metabolized by 2D6 to an active drug form, 4-hydroxytamoxifen. So this is actually what's causing the efficacy. And what they've found is that depending on your mutational status of 2D6, you can have either higher production of the metabolite or lower production. And what they've shown now is that 2D6 phenotype actually correlates with disease progression. So this is an example of variable metabolism impacting efficacy. And codeine, I think everybody's probably familiar with codeine, gets metabolized by 2D6 to form morphine. And again here we have the variable mutational status on 2D6 causing differences in safety and efficacy. If you're producing higher levels of morphine, you're at risk for morphine toxicity. Whereas if you're not producing enough of the active drug, you're actually having poor efficacy. So I think as campaigns progress, especially in CNS, if you can design around 2D6, that's even better, just because of the impact on bioavailability, safety, and efficacy. But that's something we'll talk about a little bit more detail here. So bioavailability, it's a big deal. Hopefully I've demonstrated that a little bit here. The key then for your discovery campaigns is how can you find compounds that are bioavailable? And we have mentioned that you can do in vivo testing, quick screens, but you don't have to do that just yet. This comes back to molecular properties. And essentially, the physical properties of the drugs are what's influencing bioavailability. Solubility, lipophilicity, size, these are some key features of the drug that will ultimately impact ADME outcomes. So these features will impact how the drug is penetrating across the GI tract, whether it remains soluble in the blood, how it will be metabolized by the CYP enzymes in the liver, and even how the drug gets eliminated. And the graphic on the right here just demonstrates that if you attempt to modify just one feature of the molecule, it has other impacts on other properties. So it's very difficult to purely design something and say, well, my log P is too high. It's hard to go back and just change that and not have any impact on these other properties. So they're all very interconnected, if you will. And we were fortunate enough to listen to Dr. Lipinski's lecture yesterday. I think he briefly mentioned the rule of 5, and I'll go into a lot more detail here about why it is so useful and how these types of properties guidelines have been dramatically helpful in improving attrition rates. So the rule of 5 is a mnemonic of 4 rules. And what Dr. Lipinski and his colleagues at Pfizer observed was that you were more likely to have problems with absorption and permeation if your compounds exceeded these guidelines. And now this really emerged in the 1990s as a way to improve attrition in drug discovery. So at that point, failure rates due to poor bioavailability were about 30%. So of all the compounds that we're trying to advance, about 30% were just failing purely because of that. Enlisting these kinds of guidelines reduced that significantly to today, where it's about maybe 5%, broadly speaking, outside of CNS. These are 2 examples of a rule of 5 compliant compound, Gleevec, which is a kinase inhibitor, and ritonavir, which is used in HIV therapy. You can see that this molecule here on the bottom has a higher molecular weight, higher number of hydrogen bond donors, and nitrogen and oxygen atoms. Now, one thing I should mention is that the rule of 5 is not CNS specific. This is a broader assessment tying to bioavailability. And in your own CNS drug discovery adoption of properties guidelines, some of the properties that you probably want to focus on most are log P, which is the lipophilicity, or the solubility of a molecule in octanol relative to water, molecular weight, which is size, polar surface area. This reflects the amount of nitrogen and oxygen atoms in the molecule. And we'll first start off with, how do you evaluate these? There's 2 ways to go about this. You can evaluate things experimentally. I've listed just a couple of vendors that I've personally used in the past that have been pretty good in terms of turnaround time and cost. You can also just do these assessments in your own lab if you have the proper equipment and setup. I would argue strenuously to use other services only unless you have some grad students that you can use for a couple weeks to set up systems and whatnot. But experimentally, it's not a difficult process to assess lipophilicity and solubility, these kinds of features, which can help you as you progress through that discovery engine that Marty was talking about. Now, another way to go about it, I should back up for a second. Experimentally, you're going to need a sufficient amount of compound to test that. So if your only reagent in the lab is in total 10 milligrams, experimentally determining these properties is probably not going to be an option for you. So an alternative is to look at computational analysis. I think software tools have really evolved over the last few years. Lipophilicity calculations are pretty spot on for a variety of chemotypes and scaffolds at this point. Solubility estimates are a little trickier. That might be something you want to evaluate or compare experimentally, but these at least provide a good first approximation. So prior to purchasing or using a screening library, this can be a quick way that you can evaluate whether or not it is going to be a CNS relevant or friendly library. And so it's important to kind of mention what we would think about as good values. And I struggle to put out the word good because I'll mention this again in the talk. I think these are guidelines. These are not rules. And you should always, if there's a compelling rationale to make something, don't say we're not going to make it because of the CNS rules or the rule of 5. But these are, again, guidelines to help you frame your efforts. And I'll talk in a second where we derive some of these CNS rules from. But essentially, CNS drugs occupy a more restricted sphere of molecular property space. And Dr. Lipinski mentioned yesterday, chemical space is not evenly distributed. So you want to try to zone in on the most relevant sphere of CNS space. And property guidelines like this can help you do that. I'll also mention that depending on the stage of your project, that could also impact what kind of guidelines you choose. Dr. Bahana will talk about this in a little bit more detail here, but if you're earlier in the process using a fragment-based approach, you would even want to go more restricted than the CNS guidelines. And that's because as you evolve through discovery and you advance, you're going to be adding more mass, adding more lipophilicity to your compounds. And so, as I mentioned, the yellow box, the CNS rules, this was one way that we kind of analyzed data to kind of hone in on those. What we did here was compare a database of CNS compounds relative to marketed drugs. And historically, there's been a bias that lipophilicity, if you design something with a high amount of lipophilicity, it will get into the CNS. And what this analysis shows us is that that's really not the case. There's not a big difference between the lipophilicity of CNS drugs versus marketed drugs. And in fact, as you increase lipophilicity, you're increasing your risk of CYP metabolism and efflux by P-glycoprotein. Interestingly, one of the properties that we found discriminated the most was polar surface area. So you can see that if you're designing a CNS drug, you want to try to make the polar surface area lower to improve your odds of getting into the CNS. And so this is an example where we applied properties guidelines to a CNS discovery campaign. And this box on the top is kind of just a depiction going from all the possible choices that we had to begin with. Chemical space is, consider it infinite, essentially. And the question for us being in an academic laboratory was how are we going to design molecules with only a few chemists and only a few biologists to screen them? So at the first stage, we selected a scaffold, and we limited ourselves to modifying only a couple positions on that scaffold. So that immediately contracted the number of possibilities. But still, if you just limited yourself to only 5 amines at this position and 18 different aromatic groups, you were still looking at potentially making over 1,700 different compounds. Now, if you only have 2 chemists in the lab and you really need to get through your project as rapidly as possible, you need to find a way to dwindle that number down. And the way that we began to approach that was by incorporating these kinds of guidelines. And when you analyze the properties of these possibilities and apply the criteria here, you quickly filter out a lot of compounds that you can cast aside. Now, this isn't saying that if something falls outside this guideline, you can't make it. If there is a rationale to make it, you can certainly go back. But we focused our efforts first on compounds that fit under this guideline. And ultimately, using that recursive engine that Marty talked about, we were able to go from start to lead the clinical candidate in under 100 compounds. And this was certainly something more feasible that we could get through with only a couple of chemists in the lab and a couple of biologists. And the reason I included this is simply to reiterate the fact that this is I don't know if this is actually showing up very well, but it's a poker table. And essentially, properties guidelines are really nothing more than, I don't want to say a card counting trick, it's not that, but it's simply knowing what moves to make when you see your opponent's hands being dealt out. And this is simply a way to improve your odds. I'll go through this pretty quickly, but this is just another example of a lot of these properties efforts have been purely focused with the endpoint of being CNS availability. But there are other outcomes, as I mentioned, 2D6 metabolism is something you want to avoid. And we attempted to perform some basic properties analyses, overlaps, if you will, to see what properties were correlated with 2D6 substrates and what properties were correlated with CNS penetration, to see if we could refine our guidelines a little bit. And essentially, this is a map of properties, polar surface area, and lipophilicity for CNS compounds and 2D6 substrates. And you can see that if you had followed our original guidelines, you'd be making compounds in this box, essentially, but you'd have a lot of probability of making 2D6 substrates. So if you can even narrow your properties a little bit more, you still retain your ability to make CNS penetrant compounds, but maybe you decrease your chances of making a 2D6 substrate. And subsequent work by a few other groups have now attempted to do these multidimensional properties analyses to help better refine the CNS space. And I think this is really the direction that the field is going, to kind of integrate these multidimensional properties analyses. So bringing it all together, how can you use the properties guidelines in your own work? I think first and foremost, the best application for you is going to be in library screening and selection. There are a lot of libraries available these days. You want to make sure that whatever you're evaluating is CNS relevant. Focus your efforts on the libraries that are most relevant in terms of how they fit with properties for CNS drugs, but also the stage of development that you're at. And I just mentioned for hit-to-lead refinement, it's easier to add mass and to add lipophilicity if you need it. It is more difficult for your chemists to take it away. So start low, and you will expect to increase as you proceed. And that's why applying these guidelines can help you a little bit in that regard. Your chemists may say, I can't design things that are under log 3, or I can't design things under log 4. And it's going to constrain you a little bit, but in the end, hopefully it'll help you progress quicker. And I guess the most important thing to mention is that these are guidelines, not rules. As always, don't get tripped up by the guidelines. If there's a compelling rationale, always go ahead and make that compound and test that hypothesis. And thank you very much for your time.
Thank you so much.
Oh, thank you.
One thing I don't understand is you mentioned, for example, in the rules for better CNS drugs, polar surface area being the sum of nitrogen and oxygen, I think you said?
Yes.
And then molecular weight and log P. So I sort of get molecular weight in terms of crossing the blood-brain barrier, I guess, and just in terms of passive movement, perhaps.
Yeah.
And maybe log P because of a similar reason, lipophilicity, right?
Mm-hmm.
But I don't understand why, if you have more, or if you have fewer nitrogens and oxygens, that makes a better CNS drug.
I think it relates again to, the lipophilicity, the charge, if you will, of the molecule. So these are the surface area of the nitrogen and oxygen atoms. So depending on, it's not necessarily also just the amount of the number of atoms that's present, but it's also the surface area as they're represented. So something that's more charged, I think, is going to be a little bit more difficult to penetrate passively across the barrier. And I think maybe Dr. Lipinski can.
You want to stand up, please?
Usually the way polar surface area is defined, it's the surface area of your molecule, so the solvent accessible surface area of your molecule that's populated by oxygen, nitrogen, and OH and NH. Now, why this ties in with CNS penetration is that the higher the polar surface area, the greater the interaction of your molecule is with water molecules. And the drug starts out in an aqueous environment, but when it has to cross some kind of a lipidic membrane, it has to shed those water molecules. So if you have a very, very high polar surface area, there's a strong interaction with water, and the molecule has to lose that interaction with water to get across the lipid barrier, and that's why a high polar surface area correlates with poor entry into the brain, or actually crossing a membrane bilayer.
Our next speaker will be Dr. Heather Klein. Heather will be going through the synthetic chemistry essentials of the biologist, or and black box.
Okay, can everybody hear me? All right, good morning. And I've got to say, this has been really a wonderful, the talks so far last night and today have been wonderful and really helped me set up the context of what I'm going to talk about, which is, it's a little bit misleading. There's not too much synthetic chemistry, don't worry. But it is sort of a chemistry primer for biologists in the context of drug discovery. But before we get started, I have some disclosures to make and some biases that are in the room that I feel like need to be addressed. The first being that I am a medicinal chemist by training. So that's my personal bias. And it's kind of a view of how I see the world, which is a little bit chemist-centric, so I apologize. I also want to address a bias that sometimes biologists have of chemists in this next slide, which doesn't happen too often. We try not to blow things up on a regular basis, but what I want to talk about today is really sort of a special kind of chemist, one that tries to avoid this as much as possible, and a chemist that's actually involved in the drug discovery process. And this is sort of a less detailed view of the schematic that Marty showed earlier as far as this recursive process, and we'll go into a little bit more detail of the chemist's role in this process and sort of the team role of moving from nowhere and ending up with a refined candidate. So basically you've got chemists that are involved mostly in these first steps, which are sort of finding a hit and a hit to lead approach, and then also obviously in the medicinal chemistry refinement. So that's kind of what we're going to focus on this morning. So the drug discovery chemist, obviously the foundation is you have to know how to make things, you have to know synthetic chemistry, but that's not all that is required for someone who's going to be involved in the drug discovery process. You also need to have some other characteristics such as pattern recognition and recall, and also understanding what makes a drug. And the reason I have on this slide a bunch of pictures is to point out that if you're a drug discovery chemist, you have a toolbox, things that you can use and apply towards drug discovery. And those things are translatable. They're not only for cancer drug discovery. They're not only for CNS drug discovery. They're really a toolbox that can move from area to area. And as Dr. Lipinski pointed out last night, sometimes bringing someone in working on a different area can help out because you're applying sort of that basic drug discovery knowledge to a new problem. Now for CNS, obviously, you do need to understand a lot of the things that Laura discussed in her talk is what is necessary for a drug that's going to actually be brain penetrant. But these are things that you sort of layer on to your toolbox as you move forward. So just to talk about what these things are, starting out with pattern recognition and recall and just throwing some structures up there on a Monday morning to wake you up. Sometimes you just see structures, and when a chemist sees structures, they see things in a different way, potentially, than a biologist who's trying really hard not to look at them occasionally. For example, that's TNT up here on the left. So, that's something obviously you would avoid. This is a drug that has been in clinical trials at times for cancer. When I look at that, the first thing I see is the oxygen up there, which I see can potentially break apart, may be able to covalently attach to proteins. That's how I kind of look at that molecule. Then you see things like this monster down here. It's a dye. Anything that looks like that, avoid. Okay, and if you don't know, that's part of the point of having multidisciplinary teams for drug discovery. If you have a chemist on your team, they'll say, please just move away. Pretend that doesn't exist, you've never seen it. I don't care if you think it has activity. It doesn't, just ignore it. Then you see things that are more pleasing to the eye. For example, I think Marty already pulled this out this morning, which is a derivative of one of my favorite drugs. That's, there's caffeine in there at some point. So, that's something that's a little bit more pleasing. And then you look at a compound down here, you see this is a 6-membered ring, a cyclohexane ring. It tends to be a spot for metabolism. So as a chemist, you say, okay, well, that's going to get metabolized, so can I do something? Can I put blocking groups on it? Can I block metabolism? So this is just things that, as a chemist in drug discovery, you sort of learn as you go with experience and with time. So if you have a chemist on your team, they can bring that input to you and see things in a way that you might not be used to looking at them yourself. So why do we care, right? And this goes back to what you've been hearing all morning, is really chemical space versus drug-like space. Chemical space is infinite. But if you look, this is from one of Dr. Lipinski's papers back in '04. But if you look at this huge vastness of what chemical space could be, in this green circle right here is really that ADME space, which is kind of your sweet spot. And it's not just made up, because if you look over here, you look at the log P and molecular weight that Laura was discussing, and you see that the oral drugs all really fall in this area. So you're really looking to get into the sweet spot where there are properties and activity when you're looking to make a drug. Now you notice that there are things budding out of this ADME space, for example, for proteases, some kinases. So as you've been hearing, it does depend on the context of your project and what your target is. These aren't rules or guidelines to apply in a vacuum that are the most important thing. You obviously have to think about what you're looking for, what you're trying to discover, and what you're going after in your labs. So the question is, how do you even start honing in on that sweet spot? What kinds of compounds do you use? What kind of molecules do you use? So that's when you come back to a scaffold, and Marty was talking about this. It's your core piece that you can put the Legos on the outside of, right? So these scaffolds tend to be heterocycles, and all that means is that you've got rings that have non-carbon atoms on them. And the reason, and the scaffolds are shown in blue, circled in blue, the reason that these scaffolds are used so many times in drug discovery is really driven by biology. Because the heterocycles can really interact potentially with proteins through both hydrogen bonds and hydrophobic factors. So you've got things that can form hydrogen bonds in these heteroatoms that are present. And then you've also got rings and things that can sort of interact with proteins in more hydrophobic ways. So, but obviously another key thing, if you have a scaffold for drug discovery, it has to be accessible chemistry-wise. Thank you. There we go. So you don't want a scaffold that is not able to be modified through chemistry, right? If somebody gives you a billiard ball, that's not a very good block to start with. You can't attach Legos to it. You have to think about it in that sense as well. So there's different properties that different scaffolds have. And this has been discussed both in Marty's talk, and it was discussed a little bit last night, so I'll kind of go through it pretty briefly. You look at these compounds, they're very similar to each other. There's a nitrogen that moves from here to here. So the question is, does that change the mechanism of action of these drugs, or is it sort of a smaller change, or what does it do? So this compound here is Viagra, and this one's Levitra. So actually, they're pretty similar. There's no huge drastic change that's happened with mechanism of action, but it can change the ADME properties, just the simple fact of moving a nitrogen. Some scaffolds, however, like Marty pointed out this morning, and I'll give the same example briefly, small changes lead to big pharmacological effects. So for example, in these privileged scaffolds, you've got midoprim, which is an antidepressant, and it's got some cholinergic activity, serotonergic activity. If you take this one methyl group and shift it one carbon away, then you get a completely different compound. It's got very different activity, it's got different binding properties. So as Marty said earlier, when you start a campaign, you need to understand what you're working on, understand the landscape. If I touch something, will it shift things dramatically or not? And those are things you find out through experience. So going back to this overview, the 30,000-foot view of how we go from a hit to a refined candidate, the question always is, well, where do I start? And you need to get a hit, and we talked a little bit about this last night. And there are ways that you can start to get a hit to move into this process. I'm just going to briefly go through them now. You can use high throughput screening. looking at a library, screening activity against a target or against a phenotype. You can use traditional assays. You have to have established, validated assays. You can look at patented compounds or natural products, and I know natural products will be the discussion of the next talk. And then you can look for some activity for and against specific targets. There's also a possibility of fragment screening, which is, I think, a bit more challenging for CNS, but what you're looking at when you look at fragment screening is actually a screening for binding to a target. You might not actually have an activity readout in the early stages of discovery, but you're looking for a physical interaction. So you usually use biophysical methods. So for high throughput screening, there are some advantages. You can screen hundreds of thousands of compounds, at least in the pharma setting, but when we talk about more screening like this through in a more academic setting, you're looking at the screening of a library, and Laura definitely touched on this in her talk, and I will reiterate it again. So, you might not have novel in-house libraries, but there are tons of resources out there to bring libraries in for screening. And you can use pharmacological screening and whatnot. I'm not going to address that in this part of the talk. However, whether your library has hundreds of thousands of things and you work in big pharma and it's automated, or you have a couple hundred compounds that you're screening, the disadvantages and pitfalls are actually pretty similar. And basically, what you get out is limited to what you put in. Pretty simple, right? So if you put in things that could be red flags, if you have a library that's not very robust and not very drug-like, what you get out might not help you very much. It might actually set you back. So, you might get things that are generally larger than something you would want. So you just have to be very cognizant of what you put in is limiting what obviously you get out. So, there was a paper, this is a few years ago, that just sort of discussed some of the repetitive bad hits that come out of screens. And a lot of them look like dyes that I showed in the first sign, or they have things that are very active and are not going to actually work in a biological system. They might work in a test tube, but that's about it. And once again, when you have a multidisciplinary team, you can have these discussions of balancing out, yes, I know that that was a great hit, but is that really going to be something that's a drug? It might be good for a tool in early stage discovery if you're trying to validate a target, but it's not necessarily going to be something that you could think about as moving forward towards drug discovery. So this I will go through very briefly since it's the topic of the next talk. You can also look at natural products. I just pulled out genistein, which seems to be implicated for just about everything under the sun. And if you look here, you can just look at how the change from genistein to this compound here, basically knocking off some OH groups and changing an O to an F over here, you change the cell viability, you change the cell invasion. And those are very simple, or without changing the cell invasion, sorry, but you can change the viability of the compound. So very small changes, and you end up tweaking out one property from another. So this will be the topic of the next talk, and it's definitely a very interesting area because the natural chemistry is very rich on these compounds. And as synthetic methods advance, chemists sort of have been able to use natural products in a way that really wasn't possible before. Then the next thing is a fragment-based approach. So this is finding hits based on a biophysical interaction and not necessarily on activity. So when you have fragments, they're sort of small compounds. They've got low molecular weight. They've got low log P. They've got high ligand efficiency, which is like binding energy per atom. And you're basically looking for a starting point where there's going to be a certain biophysical interaction or binding between a fragment and a target. So, and you can do this. You have a library, and then you look at biophysical measurements. So not your typical activity studies, but you can look at NMR. You can look at SPR, surface plasmon resonance. You can also use X-ray crystallography if you have, if you can, access those resources. And then you look at the fragments and you look how they bind, and you see them bind, and then you can hopefully get some sort of secondary validation through a structural method that it actually is binding. And then that can be a starting point where you can expand to get efficacy. A lot of people in past years have said, well, can we do that? And with the caveat that for CNS it's very difficult, and you really have to think about if you're looking for a specific target and how it fits into the context of your research. Going back to something that Laura said once again, there are tons of companies out there that want to run fragment screens for you, have libraries from 1,000 to 24,000 compounds in them. They'll use SPR or NMR. They have secondary technology. There's a lot of information out there. But just things to think about. Don't forget what your goal is, how these fit into the context of your project. And then also, once again, the properties of the library. Just because it's a fragment library doesn't make it better than any other library. It still needs to be vetted out independently. So this is just sort of an overview of 3 libraries: molecular weight, log P, polar surface area. And you can see they all have different distributions, different properties. And all these libraries will. So really the take-home message is for any of these approaches, you just really have to think about the library that you're getting. And pulling chemists into your team at a very early point in the discovery process is something that's really going to help when you move along to sort of give yourself the most shots on goal to get something out of it that's worth your time and money that you're putting into it. So basically, when you screen, no matter how you screen, the goal is to get a hit. So when you think about a hit, you really want it to pass certain criteria. You really want it to show potency in cell assays. If you just go from something that works in a test tube and try to move it forward, you're not necessarily going to get something that's a viable drug candidate at any point. So you're looking for a precursor to a drug and not just a ligand. You want the compound that you're working on to have chemical handles for modification, because without that, you can't run a synthetic chemistry campaign. And once again, you want these ADME properties to come through. And if you built them into the library that you're screening, then hopefully you're already a leg up in the game, because the things you get out of your library will possess these properties and help you advance things much more rapidly on the back end. So once again, the quality of the library will influence the chance of finding drug-like suitable hits. And the interdisciplinary teams are really best for hit evaluation, even if it's early stage, even if there's no actual chemistry to do yet, technically. Interdisciplinary teams to discuss the hits, think about them in the context of not just, look at that, I got a great nanomolar concentration, and there's more to it. So, and then also being able to look at these compounds that come with red flags so you can avoid them early and not get really excited about it and break your heart in 3 months when it's just toxic. So now we're going to move a little bit forward into hit to lead, and we've been breaking this up, hit to lead.